Said 1 minute.
I know, a little bit too much.
I just have to say it.
One more thing to the triple factor on that.
Then my second year from Boston. A lot of friends. It is true.
Good morning. Good morning. So let's get started. So welcome everyone to our BME seminar series hosted by Department of Biomedical Engineering at Columbia University. My name is Qi Wen, and I'm Associate Professor of Biomedical Engineering at Columbia. So today we continue to offer this lecture in a hybrid format with both virtual and live audience. So our speaker will present for approximately 40 to 45 minutes, and we will leave 15 minutes at the end for Q&A. For our virtual audience, throughout the presentation, please use Q&A feature at the bottom of your screen to submit your questions. We will try to answer as many questions as possible during the Q&A session. For our live audience, you will have a chance to ask your questions at the end of the session as well. So today we are excited to have Professor Xu Han to visit us. Dr. Han is Associate Professor at Boston University in the Biomedical Engineering Department. Dr. Han received his bachelor degree from Beijing University in China and a PhD from University of Wisconsin Madison. Her research focused on inventing novel neurotechnologies and applying innovative approaches for cellular and network mechanism of brain disorders and brain stimulation therapies. She has been recognized by many distinguished awards, including Presidential Early Career Awards for scientists and engineering, the most prestigious award for faculties, and also the NIH New Innovator Award, the Pure Scholarship, the DARPA Young Faculty Award, and Alfred P. Sloan Fellowship, and the Helen May Wiley Fellowship for her postdoc. So, okay, without further ado, welcome to.
Thank you, Chief, for the nice introduction, and it's a great honor to be here, and especially seeing all of you after this long Zoom hibernation stage. It's been good. It's on. Can people hear me? Okay. Oh, it's so great to see you all.
Sorry, I forgot my phone. I'm so focused on the audio.
All right, our faculty is amazing. Not only they can do research, they can deal with the AV issues. Thank you. I'm impressed. I need to learn more. All right, so today I will tell you mostly about our neurotechnology effort, but I'm going to tell this technology development effort in the context of my scientific interest as well. So I will be talking about calcium and voltage imaging across scales. But then in the context of this particular research topic is to use these technologies to analyze the cellular and network mechanisms for particular disease, Parkinson's disease, and deep brain stimulation. Just give you a quick outline. So I will first describe a little bit about Parkinson's disease and using local field potential oscillations as a biomarker for Parkinson's disease and potentially to use it to design closed-loop deep brain stimulation. And then I will tell you some of our recent effort on trying to understand how these biomarkers arise and what is the circuit mechanisms that gives these pathological signatures that's present in the patient brain. And then I'll tell you more recent effort about voltage imaging and then why we really need voltage imaging to understand these systems and application of voltage imaging in trying to understand the cellular mechanism of deep brain stimulation. So start with the first, a brief introduction about Parkinson's disease. Parkinson's disease is a neurodegenerative disease involving the degeneration of dopamine neurons. So here, cursor on the left is a normal, healthy human brain. The dark region is the substantia nigra. It appears dark because of dopamine. This is postmortem brain slices cut through the substantia nigra area of human brain. On the right is Parkinson's patients. Those dark region appears light because dopamine neurons die and they are no longer there. The canonical symptoms of Parkinson's is we primarily talk about motor symptoms, resting tremor, and then akinesia. They cannot initiate voluntary movement. Dyskinesia is when they start moving, they have involuntary, uncontrolled movement. And last, they have gait issues. So the circuit has been pretty well mapped, even though a lot of details are still being developed. And even this canonical two-pathway model has been challenged. But nonetheless, in the clinic, especially in the context of Parkinson's, deep brain stimulation for Parkinson's disease, this has remained very influential. So the cortex goes through the striatum, and the striatum sends out two pathways, one directly project to the thalamus, and the other one indirectly project to the thalamus. So I just want to point out a few details about this particular pathway is that in the context of Parkinson's disease DBS treatment, in 1987, deep brain stimulation in the thalamus was serendipitously found out to be helpful for tremor control. And from there, in 1997, FDA approved the use of DBS in the thalamus for the tremor symptoms of Parkinson's disease. After that, in 2002, FDA approved the DBS use of STN and GPI, these two other structures for Parkinson's disease. And since then, FDA has approved DBS use for ventral striatum areas for obsessive compulsive disorders. And there are many ongoing clinical trials for the use of deep brain stimulation. So what is deep brain stimulation? It's putting an electrode, very crude electrode, into the deep brain structures. That's why it's called deep brain stimulation. And then if you look at this figure, this red going deep, that's an electrode. The commercially approved electrode is actually about 1 millimeter in diameter. That's pretty big. And then the wires threads under the skin and then goes connected to the battery on the chest. And that battery is the same battery for a cardiac pacemaker. So deep brain stimulation is injecting high frequency electrical current into the brain. And then in the context of Parkinson's disease, it's usually about 140 hertz, really high frequency. At lower frequency, it doesn't work. And it's constantly on, and it's jamming the brain, and we actually don't know how it works. It works when we don't know how it works. So at the end of my talk, I will tell you a little bit of our recent effort on using voltage imaging to understand the mechanism of deep brain stimulation. Okay, so now we have all the patients implanted with deep brain stimulation electrode. When we are not using it to stimulate, we can record. So what we're recording are extracellular current flow. We call them local field potentials. So in this particular trace on the top is a local field potential recording from a Parkinsonian patient subthalamic nucleus, one of the nucleus that's approved for DBS treatment.
You change slides?
Oh, I did. That's I'm sorry, I didn't realize you guys are not looking at that. Why is it not working?
It was good before.
Um,
Let's try again. Share screen.
The same thing happened.
I will keep
Yeah.
an eye on this slide. I'm sorry. Yeah. All right. All right. So this is what I was. Okay. So this is what I was talking about. This wire, and then the on the top. Okay. All right, just briefly, this is a DBS implants, and then this is the local field potential recordings. And the key feature of these recordings is that it's about 20 Hz oscillations. And so that has been developed as a biomarker over the past 2-3 decades. So here is just a quick example. So this is a Fourier transform. It's not working. It's still not there.
Hide the controls. Maybe that was part of it.
Now we're back there.
Huh. It changed. It's not working.
That's a great thing. I've
Not happened before.
never been.
Oh, here, maybe it's just delayed. Now it
I'm
is.
figuring out.
Oh, it's slow. That's very slow. I'll get rid of that. Let's see. That's very slow.
She's gonna be like, I've been trying to explain it. That'll get them.
This is like where I could stop and reshare every time you have a different slide. That would be ridiculous.
But this gets the lazy
Acquaint myself with to like.
I've never
Yeah, why don't we go if it's so good?
Are you, okay, I think one option is we could just extend this.
No, it's
I
not.
think we can go full.
I'm going.
Let's have a USB hard drive.
That would work fine.
All right.
It's just a little long.
Oh, yeah.
I'm gonna go with this. I would, but I'm just. And I feel like online. Going online. I feel like it's much easier to bottle than
That's fine. That's just,
I feel like that's not. Yeah, like I never touched
for that. That should work. That should work. And then you want to get rid of the thing on the top. Watch.
Maybe, right?
Yeah. Oh, I just.
I think you'll get it.
Oh, yeah. It's like when she had last semester, I had a class at Sam Sia, and the best is when he had his back.
It's gonna be so.
Give me a shot.
Okay.
Sam Sia.
I'm gonna touch it.
Is that when it's back to us?
No, it's not. It's not advanced.
It's
Now it's working. All right, now it's working. Okay, all right. Okay. All the heads. I think I'm. All right, so I will just briefly go through this again. So this is DBS, and we can record when we are not stimulating. And I'm going to just quickly summarize it without showing some of these clinical data, is that this oscillation is there and is specific for Parkinson's patients. If you give patients L-DOPA, this oscillation is decreased. And if you do deep brain stimulation, just this quick slide, when during deep brain stimulation, we cannot record, so we don't know what happens. But when we turn the stimulator off, the patients remain tremor-free for tens of seconds. And then as the tremor resumes, this oscillation returns. So about 70 seconds later for this patient, this oscillation at about 15 hertz returns, and then the tremor returns. So these are all the evidence over the past 2 to 3 decades from these large number of patients have accumulated this evidence. So the question is, why does dopamine neuron loss lead to these pathological oscillations in the neural network, and what does that mean? So since I started my lab at Boston University, we have been looking into these issues. So here is some simple biophysical modeling. When the dopamine neurons is lost, the dopamine decreases, and the dopamine project to this striatum region of the brain, and they inhibit acetylcholine neurons. So when dopamine neurons lost, acetylcholine is increased, and that's why also anticholinergic drugs is actually effective in managing Parkinson's disease. Before L-DOPA was invented, anticholinergic drug was the only drug for Parkinson's, and it's still being used because L-DOPA has that effect. So that's our rationale. And then we found out acetylcholine can work on these M currents in the striatal projecting neurons. And so that gives us the idea, maybe it is acetylcholine causing these pathological oscillations. So we gave some drug, just squirt some acetylcholine in the striatum, and then we found the local field potential start to oscillate, generate these oscillation features, some evidence that acetylcholine may be involved. And then we want to look at what's happening to the circuitry. And this is the time calcium sensor G-CAMP was invented. And it was so beautiful for someone who's like me. I used to do optogenetics. We sent lights into the brain and we're done. It's easier. Now we have to do imaging. And so G-CAMP was so powerful and so bright for someone who has never done imaging before. We could do imaging. So this is our experimental setup. The mouse has a little head plate and is head fixed under an objective lens. And they are navigating on a floated styrofoam ball, so they can turn, they can go forward. So by reading the ball movement, we can infer the animal's movement. So on the right is this animal moving the ball, and then we can calculate the turn and the speed. And then on the right is simultaneous imaging of striatal neurons. They are green. Whenever the neuron increase their activity, they show up as red.
So is this 2-photon imaging or
This is a wide field microscope because the sensor is just so beautiful. This is wide field, so we can image over 1 to 2 millimeter area, hundreds or even potentially thousands of neurons simultaneously. And then we start to calculate how do these neurons encode, or what they are doing during movement. I also want to add another point is that because calcium imaging is green, so we can label interneurons with red with the TD tomato fluorophore. So in this case, we can image in this picture. I don't want to mess around. Okay, so there are these neurons, and then we can select these individual neurons, and so we can image their G-CAMP calcium traces. We infer neuroactivity from calcium activities. And after, while we do these activity imaging, we can also build a TD tomato map to map of these neurons. We're recording which ones are interneurons. So this way, we can, in the striatum, to look at projecting neurons and interneurons and how they interact during movement. So this paper was published, so I'm not going to go through all the details. So in this case, we recorded for the first time PV, parvobumin, GABAergic interneurons in the striatum. cholinergic interneurons in the striatum, or MSNs, also known as striatal-projecting neurons or medium spiny neurons. Those are the same name for the same neuron types. So here is just some evidence when we align to movement onset at time 0, and then the blue curve is MSN neurons. They increase in activity, no surprise. And then we found out that PV interneurons is much more powerful. They care about movement a lot more. And the cholinergic neurons, they are really crappy with their movement response. Sometimes it's there, some neurons are there, sometimes they're not. It's very interesting that we're still exploring what's happening. And then when we look at, what is happening with cholinergic neurons, we think, when we align to the peak of the movement, and then after that, the movement gradually decreases, we think the cholinergic neurons in particular is actually ramping up. So that's so basically, we think the interneurons are more powerful in their movement-related response, and the cholinergic neurons are crappy movement encoders, and the projecting neurons, they care about movement. So next, we asked what is happening to these interneurons and how they regulate the circuits. So we integrated Cholinergic activation experiments. So here, the mouse is running on this treadmill. So now you can see how they look like. And then the blue light is used to image calcium, image neuroactivity. And then red light is driving Cholinergic crimson, so we can activate the cholinergic neurons in the striatum. So now, if you pay attention, when the red light comes on, the mouse gradually stops. It's not like a sharp stop, but it gradually slows down and it stops. And so when we look at what is happening in the circuits, we realize that cholinergic activation increase the synchrony among the projecting neurons. So what we are looking at, the blue curve on the little left, is the MSNs or the projecting neurons. They tend to synchronize. When one MSN have activate, there are always some other ones. So that's the basal level of synchronization. And when we turn cholinergic neuron on, and that is the green curve, during the 635 nanometer laser, we see drastic increase in MSN synchronization or coactivity. And this is accompanied by a reduction in movement speed. And then we also activated PV GABAergic interneurons, and that's the orange curve. We found out that MSNs all suppressed. This is probably from feed-forward inhibition, very powerful inhibition. So with that, I'm just going to quickly summarize the finding of this study is that the striatum has spatial clustering during movement of different MSNs, and then PV interneurons are very powerful in encoding locomotion. And if you take one PV interneurons, they are equivalent to tens or hundreds of projection neurons. And then cholinergic neurons are just very loosely related to movement. And when they are activated, they seem to synchronize MSN activities and stop movement. And so this makes sense if there's too much acetylcholine of cholinergic neurons activated that could contribute to the akinesia of Parkinson's disease. So next we want to look, okay, how does this all related during a low dopamine Parkinson's conditions, and how is this all related to local field potentials? So that's the second part. The model we use is a neurotoxin model in rodents. This model is one of the rare cases. This Parkinson's animal model was actually first established in humans. So this is a 1980s finding in the San Francisco areas. There's these young adults, healthy young adults, developed Parkinsonism. And then the CDC went to investigate and found out a compound called MPTP. It's a byproduct of street drugs. And so this MPTP can pass the blood-brain barrier, selectively destroy dopamine neurons, and then cause Parkinsonism in humans. So we use a similar compound, but not MPTP. We use 6-hydroxydopamine because this one does not cross the blood-brain barrier. So we inject it directly into the brain. So the students and me, we are safe. We are not exposed to this MPTP that could potentially cause human Parkinsonism. All right, so we inject it directly in the mouse brain. We reduce or kills the dopamine neurons. And so in this particular case, we looked at healthy conditions, we gave Parkinsonism, and we tracked them over multiple days, and we did the same kind of calcium imaging, and simultaneously we recorded the local field potentials. And so in this particular study, we recorded 16 mice over 200 imaging sessions. We analyzed over 32,000 neurons. So that's the beauty of calcium imaging. Each time you can record hundreds, and you can do chronically over days. So that's just a really beautiful system. I just want to highlight the power of imaging. I'm not going to have a lot of time. I just want to tell you one key finding, which is really surprising to me. So striatum, when we talk about the brain, we say, oh, this area cares about vision. This area cares about motor. It's because these neurons respond during that process. What about coactivity? When two neurons are simultaneously active, what is happening? I'll tell you the conclusion is that they actually do the opposite thing, which is really surprising. So how do we analyze this? We take two neurons, traces, we calculate cross-correlation. So for example, on the left, we have projecting neurons. We calculate their coactivity by simply calculating a Pearson correlation. And then we can look at a projecting neuron and the interneuron on the right. So what we found is that when we look at the cross-correlation of these neurons, we plot it over the 200 some sessions. On the x-axis is the correlation coefficient. On the y-axis is how much they actually moved during that session. So the plotted is low speed, the percentage time, actually it's not percentage, it's actual time. 100% of the time, the animal is not moving. That's on the y-axis, 1.0, okay. So what we found, if the correlation is high, the animals are mostly not moving. So that means when these neurons are active and they're active together, they are inhibiting movement. Whereas if you have single neurons, they promote movement. So this really highlights, and then the same way for rotation. They don't move forward, they don't rotate. And then during Parkinson's condition, so we separated, this is a normal, the correlation coefficients between 0.1 and 0.2. When they lose dopamine, the correlation drastically increased up to double that. So when we look at the normal correlation coefficient, and then we found out during Parkinson's condition, they rarely move, and they are highly correlated. And then this happens right after we destroy the dopamine neurons, or we may make a chronic condition when the dopamine is chronically lost. So the point is that it's really interesting. It really makes me think, if we just record one neuron, then we say, this is our perspective over the past 50 years. This is a visual cortex neuron because this neuron responds to visual stimuli. This is a motor neuron because this neuron responds to movement. But it really depends on how this neuron responds relative to other neurons. So this was the most surprising thing from our study. Okay, so moving on, then when we look at, okay, what does this mean with the local field potential? Now we can see local field potential, we can image hundreds of neurons. So but calcium imaging is very slow. So what we did is we aligned local field potential power spectrum, their power spectrum, to the onset of calcium event. So what we see in time zero is when a projecting SPNs are active, and then we see there's not much power, but there's a little bit of an increase all the way from 20 hertz to about 100 hertz. But then after dopamine is destroyed, on the SPN onset, the lower one, we found this massive power at the high frequency. And then we also know when movement occurs, neurons active. So on the top, we find out, well, when movement is happening at time zero, there's not much change. There's some neuroactivity. They're broad and they're just non-specific, and it's lasting for like seconds, 2 seconds. Whereas this transient, we want to highlight this. I know the people can't see online, sorry. So this is a very transient, lasting only about 200 milliseconds. And this is pathological. And so this is called high gamma oscillations. Forget about the Greek letters. But it's really high frequency. And this actually occurred in the motor cortex in Parkinson's patients. So this was interesting. We really wanted to see this 20 hertz oscillation. That's the biomarker, but we couldn't find it. We looked, we don't know what's happening, but it's not there. We didn't capture it. But instead, we captured this massive increase in higher frequency oscillations, and they increase during the dopamine depleted states. both acute and chronic. And then when we looked, when this high frequency local field potential oscillation, what does it mean? The hypothesis is that they are related to correlated activities. More correlation, activity that leads to this extracellular high frequencies increase. So we plotted that. On the y-axis is correlation between neurons, and the x-axis is this high frequency, we call it high gamma power. The more correlation, the more power. This happens in healthy condition, in acute condition, in chronic condition. So right now, we don't know what exactly is changing, but my hypothesis is probably there's these states. They are naturally occurring, and then when dopamine is lost, the state is highlighted or exaggerated, and then the movement, the mice just don't move much. That's causing akinesia. Just to summarize this part, we did have some evidence that beta oscillation, 20 Hz oscillation, is associated with movement deficit, even though we couldn't find a particular network feature that goes with it. And then we found elevated SPN projecting neuron synchronization after dopamine loss. And then we also found that these local field potential oscillations does have some kind of network features that's associated with it. So at this point, the take-home message for me, and I hope I explained clearly enough, is that, okay, single neurons are interesting, but it really depends on how they activate relative to other neurons. And then what about spiking? We're talking about calcium. And what does it all mean? What does these oscillation transient, like 10, 100 milliseconds, really relates to neuroactivity? Calcium is very slow. I can't answer that question. So we really need voltage imaging. So now I hope I convince you guys why we need voltage imaging. So here, this is the beautiful study of GCAM. Each spike at the bottom is associated with a beautiful calcium spike. But if you do calcium imaging, you know that's not a true story, because it really depends on the cell type and what we are looking at. And so this is a new study by the Allen Institute. They did surveyed a lot of the cells, but a lot of the times the spike is not associated with the calcium spike. And the calcium spike sometimes occurs without action potentials. And so what we really need is voltage sensors. So voltage sensors are hard. Why is it hard? For those of you who don't think about these sensors, I'll just point out a couple things. Calcium is intracellular. You can put a lot of a sensor and stuff them in the cytosol. There's a lot of real estate. And then you count photons. There's a lot of sensors. You can count a lot of photons. When we talk about voltage imaging, we're talking about the membrane. Everything needs to be on the membrane. They're very limited real estate, very small area we can put our protein. That's why it's hard, because the number of sensors we can put in a neuron is very limited. And now we require them to also sense at sub-millisecond time scale. The number of photons we can count within a very short time period is also very limited. So that's why voltage imaging has been so hard. So here, fast forward, people have been trying to do voltage imaging since the 1980s, 1990s, and there hasn't been really great sensors. So here's one. It's a mutant of archaeorhodopsin. For those of you who have studied optogenetics, this is the very same archaeorhodopsin that I worked on and developed as a postdoc. This is the arch that you use for optogenetic silencing. It turns out these rhodopsins have these very interesting optical features, and they have a near-infrared sensing of fluorescence that's dependent on voltage. And so over the years, started by Adam Cohen's group, they have mutated the proton pumping capability of this molecule. They enhanced the voltage sensing capability. And then after a few iterations, this particular one is called ARCON1. This was developed by Ed Boyden's group, that they have done a large-scale robotic screened thousands of mutants, and they found this particular one. So here is a characterization. The gray curve is the classical patch clamp, and the pink curve is the fluorescence of ARCON1. So you can see they are just perfectly matched. And of course, this is in vitro, and I'll show you some in vivo studies. And then not only that, the fluorescent change is linearly related to the absolute membrane voltage. So they published this paper in 2018. It's called ARCON-1. We were so excited. We got the sensor, we put it in the mouse, we cannot get anything. The reason is that neurons, they have a cell body, they have the processes. And then when we talk about kilohertz imaging, we have to rely on wide field. And then there's a huge amount of background. We don't know, we can't see the cells. So that's a killer, we were disappointed. And then what we did is that, okay, let's do molecular engineering. You know, optics, you can make 2 photons faster or some confocal faster, but a kilohertz is not easy. So we molecularly restricted the expression of ARCON-1 to the cell body. So now that's the middle. So we eliminated the background using molecular engineering.
Slide over a little bit to the left.
All right. Okay, so that's for, so this sensor, we call it soma targeted ARCON. So we put it in the mouse brain, and this is the exact same setup I showed earlier as calcium. The same setup, except we replace the blue LED with a near infrared laser, and the mouse is still running around like I showed earlier. So we tested it across multiple brain regions, motor cortex, visual cortex, striatum, hippocampus. The short story, they all work. So I'll just show you one example. This is a single hippocampal neuron in a awake mouse, kind of doing the same navigation, ball moving thing. And then this is a trace of soma ARCON fluorescence. I zoom into the pink area, and I'm zooming in a little bit further. So now we can see individual spikes, and this is collected at over 800 hertz. Why 800 hertz? That's the fastest this current camera can run. This is a field of view. We just let the camera run as fast as possible. I think it was 226. And then this is the data we get, and we can calculate how good our spikes are. So this is example. Here is a video. I think you can see the flashing. So this is collected at 826 hertz, and I'm playing it at about 20 hertz. The bottom is the fluorescence of the cell body. All we did is some motion correction. There's some micro motion. And so it's pretty easy to use. It's just a classical custom wide field microscope. So with that, this molecule is near infrared fluorescence, so we can couple it with blue light optogenetics. So this is an in vitro study. Each blue pulse on the top is a pulse of light, and this neuron expresses both the voltage sensor and also a channel rhodopsin. And so now each blue light drives the channel rhodopsin and evokes a membrane voltage change, and that membrane voltage change is measured with soma ARCON. It works beautifully. And then when we do this in vivo, the blue light is showing the activation of this neuron, and the spike is increased. So we can do everything all optically, and we can do multi-color control. So here is an example of the striatum. Let's go back to the striatum. On the top is a neuron we imaged. We see spikes. I really want to use the pointer. Okay. There.
Yeah, it's good.
Great. And so here is the spike, and then the bottom is the movement velocity. So in this case, when the movement is a little bit lower, we can see the spike, this neuron is inhibited a little bit. So this is the first time we are actually recording single cells in vivo during behavior using voltage imaging. And we characterize these neurons, and then we can do all this. Some neuron go up, some neuron go down. That's just classic system neuroscience. And then we can see their areas, and we can do all these things because we're imaging them. So this is nice. And then we can use genetic tricks. So this is actually the cholinergic neurons. We put these voltage sensors selectively into cholinergic neurons in the striatum. They are traditionally called TAN neurons, tonically active neurons. So, but you know that's just using an extracellular electrode. So we actually, the features is quite different. So we're really excited. We're looking at these neurons. Some burst, some don't. So this is an opportunity. We can look at these. And then what about subthreshold? Because if you do, when we talk about oscillations, local field potentials, what are local field potentials? So there's a lot of debate because we don't know. It's extracellular current flow. Is it action potentials? Is it synaptic potentials? A lot of people believe it's synaptic potentials. Action potential is less than 1 millisecond. It's very, very sparse, and it's very local only at the cell body. But synaptic potential is slow, it's massive. So a lot of people believe local field potential measures synaptic potential. And so that means synaptic potential are mostly subthreshold. They do not drive action potential. So we think it's gonna be subthreshold. But I can't prove it because you have to do a lot more experiment to prove this. So what we looked at is that, okay, what about subthreshold? So we compare the local field potential we recorded and the neuron we imaged. And then in the hippocampus, there's this oscillation. It's called theta oscillation. It's about 5 to 10 hertz. It's broadly documented. It's involved in learning and memory, navigation, and the organized spikes. So there's tens of years of work on this. So we filter the local field potential in blue, and then we filter them and look at the subthreshold oscillations. And then this is the neuron, a single neuron, and we filtered it, and then we got their oscillation, subthreshold oscillation. So when we look at spikes, so we see the spikes occurs on the top, on the peak of the subthreshold oscillation, and their relationship to the local field potential oscillation is very heterogeneous, it's not tight. And so this is interesting. So we found, okay, we characterized it. Basically, the phase relationship of the spike relative to local field potential oscillations is much weaker to their own oscillation. There were a couple studies did patch clamp recordings in awake mice, and they saw similar results, but they only recorded a few neurons. So here we can record a lot more neurons, and this just demonstrates that our sensor can detect subthreshold dynamics, and these results are consistent with the gold standard classical patch clamp. Okay, so I think I have about 20 minutes.
Maybe if you could finish about closer to 10 or 15, it'd be better. So
Okay, sounds good.
We need to end at 12.
Okay,
Sorry.
sounds good. All right, so wide field has a lot of background fluorescence, so we built a pattern illumination system, very simple system using digital mirror devices. I'm not going to go through all the details. I'll just show you some data. So this is in cell cultures. We can record tens of neurons simultaneously. I did patch clamp as a grad student. I know how annoying and difficult it is each day, and you usually can't get many. And so here I can see tons of them just in a single snapshot. And then you can see in culture it's so easy. And then the students in my lab doing in vivo recording, when they see this makes them want to cry. this is so no artifact. And you can even see these gradual, depolarization that drives the spiking. It works great. My point is the sensor works great. In vivo, when we did the pattern illumination, this is in the visual cortex. We can record continuously, in this case, 5 minutes. Of course, there's some photo bleaching, but still the signal remains pretty good. And I just want to highlight this. This is in the hippocampus again. When we have beautifully labeled neurons, pretty dense, then we can image and pick pattern that we want to illuminate. So for the first time, we achieved 76 neurons simultaneously imaged in vivo during behavior. And of course, we recorded it for a few seconds, and many of them spiked. And so this was with at that time, about 2 years ago, the best camera on the market is a Hamamatsu camera. After that, we found out this really beautiful camera. It runs even better, even lower noise. And not only that, we can drive it at 10 kilohertz. Of course, when we do 10 kilohertz, we're really only looking at a single neuron, so that's there. So this is a 10 kilohertz imaging. So what we found is that in the hippocampus, there's these spikes. We can get this spike, and then there's a cluster of spikes that rise on top of the subthreshold depolarization. This is called complex spikes. It's very common in the cerebellum. It's also in the hippocampus. And a lot of science physiology studies have characterized, okay, there involves calcium channels, and the action potential actually broadens. It becomes broader. So the question is, can we use SOMA-ARCA to capture real action potential shapes? So here, of course, it's not as good as patch clamp, but then you can see even in this example, the action potential got wider in a sequence of these complex spikes. So we characterized it when we record at 10 kilohertz, 5 kilohertz, and 1 kilohertz. 1 kilohertz, of course, is too slow. So we did confirm that we can capture action potential widening. So the half width the full width at the half maximum amplitude is less than a millisecond. And towards the end of this fifth or fourth spike, they actually double their action potential width. So we confirm, even with the sonar archon, this optical methods, we can capture action potential shapes. So now we can really start to think, are all action potential the same? So that's the question, right? If they are broader, does that mean I'm going to release more neurotransmitter? Is it going to be more faithful in synaptic transmission? So these are all the questions that will be really interesting to look in the future. So this is just another example in the visual cortex at 5 kilohertz with sonar archon. So I just want to summarize this. This sensor can measure both action potentials and subspatial dynamics. We can record from specific neurons, and we can do optical control and imaging. And then we can do large scale, tens of neurons easily with a very simple patterned illumination microscope. We can do super fast imaging at 10 kilohertz. All right, I think I have about 5 minutes. I will just go very quickly of how we have used it ever since we got this sensor to do some science. Okay, so deep brain stimulation. I like this paper. This was written by Benedict. He is a neurosurgeon that discovered this DBS for Parkinson's patients. And he wrote this review in 2009. And then he wrote this equation. As engineers, we all like equations. So it's a function of mechanism times A1, and so on. And so all these A1s and Mach1 is all unknowns. The point is, there are many, many mechanisms that could contribute to the DBS therapeutic effect. We were laughing at it. Engineers, you all want to write this equation. This is interesting. But the way is that we don't know how deep brain stimulation works. The reason is we record electrical activity. And then here we're stimulating at 140 hertz that creates electrical interference. Then we really don't know what happens to neurons during stimulation. A canonical hypothesis based on neurosurgeon observation is that if you do DBS, patient get better. Now, if I lesion the same area, you do DBS, the patient also get better. So the rationale is, okay, DBS and lesion are the same, so DBS is creating an inhibition. Is it really creating an inhibition? We really don't know. So here, what we did is we did the same voltage imaging, but in this case, we went to the hippocampus again. We put an electrode, and we delivered electrical stimulation. We imaged that neuron. So this is a 40 hertz deep brain stimulation versus 140 hertz stimulation. I just want to point out here at 140 hertz, we see membrane depolarization. The membrane voltage depolarized, and it stays out, and we do not see action potentials. Why? Maybe the neurons are leaky. Maybe they are blocked. We don't know, but the spikes are inhibited. When we do 40 hertz, we have some depolarization, but it's not enough to inhibit the neurons, so the spiking is crazy. So that's probably why deep brain stimulation really need to be high frequency, because in patients, it's always high frequency, about 130 hertz. So maybe this is something interesting. So in some cases, we definitely block them. And then when we look at the magnitude of depolarization, 140 hertz is very fast, it's depolarizing. When we look at the spiking, how many action potentials are still occurring with 140 hertz during the stimulation, they're still there. 140 hertz, in the beginning, we have a transient increase and it quickly decreases. And then it's not inhibited completely. It's still there, it's just random now. So we unfortunately didn't see complete inhibition. There's some escaping of action potentials. And then what is going on, in addition to this, so we did optogenetics and a DBS and voltage imaging. So we're doing DBS, we're driving this neuron with blue light. So we ask the question, if I have an input, I'm driving the neuron, and then the neuron going to respond. Now with DBS, is the neuron going to respond the same way? So here, before DBS, each blue light pulses drive the neuron spiking beautifully. During DBS, all the spikes are scrambled. They are not following the inputs. And so this happens, so we zoom in. Without DBS, the neurons can follow input. With DBS, the neurons are just not following input. So basically, we characterize this when we do 140 hertz, the neurons are also not following inputs. So basically, the neurons are no longer capable of following input in the presence of DBS. And so we can characterize this. I'm going to just skip that. So we characterize the amount of following to the input versus the amount of depolarization DBS created. And we concluded basically the more depolarization we have, the less following. So it's the suppression of this following of the inputs. So with that, just to summarize, we think DBS strongly depolarize neurons. So they are depolarizing neurons. We observe this at both 40 hertz and a therapeutic 140 hertz. 40 hertz can entrain spikes, and then the spike just occur follows the electrical pulses. 140 hertz, we see very little of that. But we still see spiking. So we didn't see complete inhibition. Okay. And so we also found that both when we gave electrical stimulation, we found the neuron's ability to follow input is reduced. So their information transferability has been impaired. So we are very excited by this study, and we really want to do more of that. With that, I'll just thank people in my lab, my collaborators, my founding, and many alumni and undergrads. On the calcium imaging side, that's on the left, on the voltage imaging side, these are the people contributed to the data I showed today. I'm not going to read out their name because I'm a little short on time. So thank you.
Thank you so much. Where's the rest of the script? You want me to read it? Okay. Got my talk here. Okay. Thank you so much. Before we get into the Q&A, I just want to call everyone's attention to our next hybrid seminar, which takes place next week, Friday, April 8th. We hear from Professor Vali Malini from Tufts University as she presents multi-parametric nanotechnology-enabled ultrasound-guided photoacoustic imaging for treatment guidance, dosimetry, and tumor recurrence prediction. And we also want to remind you that on April 23rd and 24th, Columbia Bioengineering is hosting the 48th annual Northeast Bioengineering Conference here in New York City on our Morningside campus. Here, you can see it on the screen here. Check out our website, nebec.org, for more information, and registration is now open. Okay, now is my favorite part, which is the Q&A. We got that out of the way. There are a number of questions online, but let's start with the ones here. The catch.
Hi, thank you for your presentation. I worked with calcium imaging as well, and I was wondering if you could expand on either the signal processing or algorithmic procedure you use to isolate the signal coming from one neuron to another. Because in the data I have from similar technique, you have a lot of like overlap. You have a lot of diffusion of the light. So how did you like eliminate that?
Excellent question. Yeah, so for wide field background contamination is the issue. So usually we can do a subtraction. So we take the cell body and we subtract the surround area. So that reduces some of the background. We also worked with a SOMAR targeted GCAM in collaboration with Ed Boyden's group that molecularly restrict the GCAM into the soma, so that drastically reduce the background fluorescence. So I think that's helpful. It's also interesting, we compared when we do signal processing using the soma minus the surround, the surround is the estimation of the background, and then do cross correlation between neurons, and that achieved about the same as we use these molecularly target sensors. So I think the take-home message is that you can do some signal processing by reducing minus the surround background. It's not absolute. And if you do a molecular targeting, that's it's a better targeting because you don't have fluorescent, you still have some, right? So I think, unfortunately, with wide field, you just have to work with these signals. In the targeted illumination system, we estimated with SOMAR-RCOM is that if neurons are sparsely enough, and they are more than 40 microns away, their cross-contamination is drastically reduced.
Hi.
It's on, right?
I don't think.
It's on.
First of all, thank you so much for the presentation. Very exciting science, honestly. I was just wondering, for your arc on, have you looked into making like a Cree-dependent version of it?
Oh, we have it.
You have it.
That's
okay.
how we use to measure the cell type specific ones.
I see. Okay.
Yeah.
Mhm.
And it's also online. You can get it from Edge.
Okay. Thank you.
Thank you as well. Have you done any work doing SELMA or Hillock-targeted channel adoption, or would that be unnecessary?
Cell targeted channel adoption.
Like SELMA or maybe Hillock-targeted?
It's already, you mean the cell targeted channel adoption or optogenetic?
I'm saying targeting channel adoption to be concentrated around the action of Hillock, so you potentially use less virus to.
People have targeted channel adoption onto the soma that's published. Specifically to the axon Hillock. Here's the story I heard. I don't know whether the paper is published. Because if you put channel adoptions highly concentrated on the soma, it knocks away the sodium channel. Then the neurons will spike at him. I think it's too much interference. I heard some people talk about that. Yeah.
That makes sense.
It is a good idea, but then there are other side effects. Yeah.
All right, why don't we take some questions from the online audience here? Mian Wei had a question that came from the beginning of the talk. What is the imaging depth of voltage imaging, and why is it possible to image CA1 neurons using wide field microscopy?
Excellent question. Unfortunately, wide field, we are imaging somewhere between 50 to 100. Maybe we can push to 150 microns from the plane. So to get to the CA1, we have to take everything above it out, so surgically removed. So that's pretty much true for calcium imaging or anything with wide field. That's unfortunate. Yeah.
There's another question from Ian Zheng Kwan. Can you explain what instrument do you use viral injection to label calcium?
Oh, that's just, we just use a micro injector, and we do it either before we put on the chamber. We also have some chamber with an injection cannula. It's just an injection, typical injection cannula.
Thank you. This is a very insightful talk, and I learned a lot from it in terms of the mechanisms of deep brain stimulation. And you have shown that DBS at 40 hertz and 140 hertz has this sort of mechanism of decoupling the stimulated or inhibited neurons from the input. Have you done any preliminary work, or do you have any hypothesis about expanding that to a larger network connection? And you have, so you're inhibiting these ones, these neurons, but those are decoupled from the input. Does that have an effect on the neurons that have input into them? And then what is the downstream effect? And can you find better targets for DBS with something like that?
That's exactly where we want to go. We have not yet obtained any results, but I think that's exactly the point, the upstream, downstream, or the network synchronization. I think this will be a really interesting question to answer. Need more people to work on that. That's interesting.
Thank you. Very exciting.
All right, we have a couple more questions before the time gets cut off on us here. Curtis DeVoe had a bunch of questions. Maybe we'll start with the first couple. Could LFPs correspond to plateau potential upstates? During plateau potentials, the spikes may be attenuated or difficult to detect spikes.
Yes, I think the plateau potential, if we're talking about instrument, I don't think from the sensing perspective, it is. But I think during that time, if we talk about mechanism, a lot of ion channels could be open, and that increase in conductance could reduce spike rate. Less, the capacitor, this is conductance too high, then you don't generate spikes, or it could potentially completely block spike. And also, if you talk about sodium channel inactivation, that's another question. that could contribute to the inhibition of spikes. There are many ways mechanistically this will lead to a decrease in spike rate. So I guess, is that the mechanistic question, or is it like instrument-wise? For SOMAR outcome, this is all, we should not, it's pretty linear up to about plus 60 or 70 millivolts, so I don't see a reason of why we will have reduced sensitivity. For local field potential, I don't know. This is extracellular current. I don't see any reason of having an effect.
Okay. And one last question before we wrap up. This was a little bit more fun, perhaps, or I don't know what it is. Are you planning on replicating these results in humans?
Oh Lord. Fascinating question. Maybe slices from epilepsy patients if piece of their brain tissue is taken out.
You're getting volunteers for live?
Can we do this? Right. Let's address the easier question. For channel adoption, people try to do human therapy, right? Going into the central nervous system part. People are trying retina, and they're on a trial on using channel adoption. For voltage imaging, I think that's way far off. I wouldn't want to do that, or I wouldn't even recommend doing that. So it's mechanistic study in animals.
All right, thank you so much. That was great.